Hello, my name is Favor and I'll be presenting on DNA binding proteins with a focus on Taq polymerase, a DNA polymerase 1. The DNA is essentially the blueprint of life. It is the molecule that contains information for the growth, development, as well as function of an organism. And the DNA binding proteins are a group of proteins that regulate activities related to DNA replication, transcription, repair, among others, and they do that by simply interacting with DNA molecules by means of their DNA binding domain or functional group, which essentially interacts with the major group of a DNA molecule. Here are classes of DNA binding protein. I'll be focusing on DNA replication factors such as Taq polymerase. DNA polymerases are enzymes that catalyze the synthesis of DNA from nucleoside triphosphates. They play essential role in DNA replication and repair. And there are different types of DNA polymerases. For example, in prokaryotes, we have type 1 to type 5 DNA polymerase. Taq polymerase is a thermostable DNA polymerase, one derived from a bacteria known as Thermus aquaticus, a bacteria that lives in hot spring. And it has a wide range of molecular biology application, for example, in application of gene or target DNA sequence during polymerase chain reaction. And it is very similar to the Escherichia coli DNA polymerase 1 in its primary, tertiary, and three-dimensional structure. However, it lacks the 35 endonuclease activity, which is very essential for the providing function of DNA polymerases. And it is used in polymerase chain reaction because it can withstand the high temperature required to split the double strand of a DNA molecule. The structure of Taq polymerase resembles a cupped human hand with various subdomains. Here is the thumb region, and here is the finger, and here is the palm region, which is the catalytic center, and here is the 35 exonuclease, which is non-functional, and at the end terminal we have the 5' to 3' exonuclease, which is functional. Taq polymerase produces single base substitution errors at the rate of 1 in 9,000 nucleotide due to the lack of 35 exonuclease activity accounting for its fidelity property. Taq polymerase works during the extension step in polymerase chain reaction where it synthesizes a new strand of DNA complementary to the template. It does that by binding to the primary DNA template complex following annealing. After that, it extends the going strand in the 5' direction, adding nucleotide following the base pair rule of adenine to thymine, guanine to cytosine. Taq polymerase has a wide range of application in disease diagnosis, DNA sequencing, mutagenesis, and gene expression studies, and here are the references.